Well, hello everyone. Today we are going to briefly review structures of DNA. We will identify circumstances when DNA must make another copy of itself. We will describe the role enzymes play in DNA replication, and we will use Chargaff's rule to build an identical strand of DNA, and that's also known as our base pairing rule. To begin, let's remind ourselves that DNA is this structure. Its shape is a double helix. And this double helix is built by individual subunits, which I am circling right now at 3 components, which is a sugar. In the case of DNA, it's deoxyribose. It's got a phosphate group, and it's got a nitrogenous base, of which there are 4 types. All right, and they alternate, or they should, I should say, they get linked together as a string, but 2 strings that are then loosely bound together with a hydrogen bond. And this here is called a nucleotide. Morning, Penny. All right, so if we now take a look at a nucleotide. So this is a nucleotide. It's the smallest subunit of a nucleic acid. And these nucleic acids are DNA and RNA at the moment. Let's not talk about RNA. But here's that phosphate group. Here's the sugar. In the case of DNA, it is deoxyribose. If we are talking RNA, it's called ribose. And here is the nitrogenous base, of which we could have adenine, thymine, guanine, or cytosine. This picture is showing multiple nucleotides strung together. On the left side, well, got a few questions for you. Let's complete the strand first. Let's go there. So pause the video, attempt to complete the strand, and then see if you were right. All right, so on the other side of C, connected to it, the complementary base is a G. Complementary base to A is T, complement to G, C, and T, A. All right. How many nucleotides are present on the screen? Pause, and let's see. So, right here, we have 1 nucleotide, which means that this is a second, a third, and a fourth. There's a complementary strand on the other side, so we have a total of 8 nucleotides. And what does number 2 point to? Number 2 is right here, and it is asking you to visualize that invisible force that's holding both strands together, the complementary strands, and this is known as a hydrogen bond. Okay. There are times in which DNA must make more copies of itself. So what are these times? Well, when you grow. To begin with, you started out as 1 cell big. How did you get to be trillions of cells big? Well, you had to make more cells. To make more cells, you need more DNA. Another time, we have to replace old cells. When your skin renews itself, when we wash, we're taking off old skin cells. Well, there must have been some skin cells ready to go. And they needed more DNA. And last, we need to repair. Damaged cells, well, they're removed. We need something to take their place. So we need more cells, we need more DNA. So we're talking about DNA replication. When DNA replicates itself during mitosis or meiosis, it makes a copy from the original, and it does it by doing the following, but pause just for a moment. The term mitosis. Mitosis means from 1 cell, from a parent cell, we are going to make 2 daughter cells. All right, and that happens with the examples I just mentioned to you. Meiosis we'll talk about later, but deep down, meiosis means that we are preparing sex cells. So we're preparing eggs, we're preparing sperm, and we need more DNA to be able to do that. So let's break this down. To begin with, this DNA, it must be unzipped. So we're unzipping it by breaking the weak hydrogen bonds that hold the complementary strands together. And to do that, we're going to use an enzyme called helicase. You'll see that later. Okay, then we have to build new complementary strand on the two existing single strands. So those strands that were intertwined are separating. This, if we build a new complement to it, will be a separate strand. This one here, build a new complement to it, will be a separate strand. So let me start this statement over again. Build new complementary strand on the two existing single strands of DNA. By following the base pairing rule, that Chargaff's rule, two identical strands are now built. And the two strands will be clones of the original. So on this slide, I'm asking that you regularly pause to read through it because I'm going to be going through it rather quickly. So the parent molecule has two complementary strands of DNA. Each base is paired by the hydrogen bonding with its specific partner, A with T and G with C. This next slide. All right, you see how they've been separated? Well, the first step in replication is the separation of the two DNA strands. Remember, I said an enzyme assists with that. Otherwise, it wouldn't fall apart. So a separate molecule called helicase, an enzyme, will unzip it. So the hydrogen bonds are now broken by that helicase. We say that it has unzipped. And now we have those two template strands. And each parent strand now serves as a template that determines the order of nucleotides along the new complementary strand. You see those light blue ones? They are rebuilding a strand. Actually, not rebuilding. They're building a new strand. So the A's goes with the T's, and the C's go with the G's. And last, the nucleotides are connected to form the sugar phosphate backbones of the new strands. Each daughter DNA molecule consists of one parent strand and one new strand. So these dark blue ones came from the original. These light blue ones are the new ones. And if you pause and examine them against each other, you'll see they are identical. And even compare them against this parent strand, you will again see that they are identical. All right, so now let's practice for ourselves. So I'm going to unzip this strand of DNA. So unzip the DNA and compose the complementary strand of DNA. So I will unzip it for you. I then want you to pause and attempt to build the complementary strands. So go ahead and pause now and see if you can do it. All right. It should look something like this. The T goes with the A goes with T, C goes with G, G goes with C, and I'll just keep on going. T, G, A, T, C, T, T, A, C, G, G, T, A, C, T, A, G, and A. So if you compare these against each other, hopefully I did it correct, and you'll see that they are identical. And here's that original. Let me put it right in between, compare them both. They should be identical. So now what I'm going to do is I'm going to show you a few, two different videos that came off YouTube, and they're a little bit over a minute each. One shows this process in a cartoon type drawing, and then the other one, quite amazing. It's a CGI cartoon, let's call it, or video, and it represents what we think it truly looks like. So let me go ahead and pull those up. Here's the first one.
DNA is a type of nucleic acid made up of 4 bases: thymine, adenine, cytosine, and guanine. Thymine always pairs with adenine, and cytosine always pairs with guanine. DNA has a shape that can be compared to a ladder. The bases make up the rungs of the ladder, while the sides of the ladder are made up of repeating phosphates and sugars. The unique structure of DNA allows it to be easily replicated. One strand of DNA can be used to form the complementary strand. Matching bases are added until the two strands are complete. There are several important enzymes involved in DNA replication.
There's that helicase I mentioned.
Helicase separates the original strands of DNA. Then, DNA polymerase adds the nucleotides to make.
All right. This other enzyme, that's DNA polymerase. Notice that both the helicase and polymerase end in ASE. If you recall when we went over enzymes, most enzymes take on a name that has ASE at the end, such as sugars are usually end in OSE.
The new strand of DNA. DNA is replicated during the S phase of the cell cycle so that the cell can divide during mitosis.
And there you go. Let me bring in another video. Now, that one was in cartoon form. This one shows you it, well, as we believe it really looks. On the left side, you'll see that that is a double strand of DNA. This one here is a double strand of DNA that is being built. Right here, they'll talk about how it's built. Well, one of the strands is built in a backwards form. Don't get too wrapped up about that. But let's see what this one says. I'll probably talk over it a couple times.
In computer animation based on molecular research, we are now able to see how DNA is actually copied in living cells. You are looking at an assembly line of amazing miniature biochemical machines that are pulling apart the DNA double helix and cranking out a copy of each strand. The DNA to be copied enters the production line from bottom left. The whirling blue molecular machine is called helicase.
So this guy right here, there's the helicase. Here is the parent strand of DNA entering. If you notice over here and over here, that's the DNA as a single strand. It has been unzipped.
It spins the DNA as fast as a jet engine as it unwinds the double helix into two strands. One strand is copied continuously and can be seen spooling off to the right.
All right, so if this molecule is assisting with rebuilding it, which enzyme is that? Yes, that's DNA polymerase right here. And there's another one right over here.
Things are not so simple for the other strand because it must be copied backwards. It is drawn out repeatedly in loops and copied one section at a time. The end result is two new DNA molecules.
All right, so just a quick review. So we went over what DNA looks like, its structure once again, so that then we could see how it is DNA will make duplicates of itself. So watch the video as often as necessary in order to understand the material, and have a great day.